In this video, I'll take you through example 3 on the Pipebio documentation site. Compare samples from a biopanning experiment. You will learn how to import FASTQ files, annotate sequences, and cluster and compare multiple samples. The example dataset is a VHH phage display library, constructed from Alpaca PBMCs after immunization with an IZ-UMO1 antigenic fragment. This is a public dataset published in 2015. To start, download the FASTQ files from the Pipebio documentation page to your local machine. Unzip the downloaded files and inspect the contents. If you are on a Windows machine, make sure to extract the contents fully. There are two samples, one before panning and one after panning. This is a relatively simple example, but it's worth noting that Pipebio can support more complex study designs from multiple panning rounds. The paired Illumina MySeq reads have already been merged, so there is one merged file per sample. See the example 1 tutorial for more information on merging paired end reads. In Pipebio, create a new folder in a project. You can import the files either by going to File, Import Files, and browsing to the location on your local machine, or you can simply drag and drop the files directly into the new folder. If metadata columns have been configured for your organization, you will see an extra dialog to set those values. If not, you'll proceed directly to upload. You can monitor the progress in the jobs table, which also shows you the upload speeds. Each FASTQ file will be in a separate document. You can open the documents to inspect the reads. And inspect the quality charts. Again, see the example 1 tutorial for more information on NGS-QC. We can annotate the sequences in a high throughput manner. Select both documents. Go to the sequence menu at the top and choose Annotate. Choose the VHH scaffold and choose the latest version of the Alpaca germline database. Click Annotate. Open the annotated sequence documents. Use the status chart to get a quick overview of the annotation results. The vast majority of sequences were correctly annotated. Again, see the example 1 tutorial for more information on inspecting the annotation results. Next, we need to find clusters of sequences across samples. For each cluster, we can quantify the number of sequences in each sample and compare them to find enriched groups of heavy chains after panning. Select the two annotated documents. Go to the sequence menu at the top and choose Cluster Multiple Samples. Give the samples shorter, more meaningful names. The 18583 sample is before panning, and the 18582 sample is after panning. Click and drag to rearrange the samples so that the baseline sample is at the top. All other samples will be compared to the baseline sample for fold change calculations. You have the option to pre-cluster on germline gene assignments. We won't do that for this example. You can generate clusters for a particular subregion or multiple subregions. In this case, we will cluster on the full heavy sequence region. The percentage identity threshold can be increased closer to 100% to ensure all members of a cluster are completely identical, or decreased to allow variability within each cluster and to account for sequencing error. In this example, we will leave it at 90%. We'll use the standard translation table and keep all the other defaults in the advanced settings. For bigger datasets, you may find the clustering is taking too long, and you may want to consider increasing the word size to, say, 5. We will leave it for 2 in this example. Also note that a filter to include correct sequences is automatically applied. Click Cluster and Compare. One output document will be produced containing the clustered sequences and comparison results. Open the compared sequence document to inspect the result. Click the charts icon to view new charts that were added after clustering and comparison. The comparison chart shows the cluster counts before and after panning, for the 30 clusters with the highest fold change. Each coloured line is a different cluster. If we remove the cluster with the highest fold difference by clicking 18571 in the figure legend, the y-axis will rescale to show the other clusters. You can select a line to automatically filter the table below for sequences belonging to that cluster. Let's use the table to explore some of the cluster results in more detail. A number of new columns were added to the table, including combined cluster information, which summarizes clustering across selected subregions, count and fold change information for each sample, and more detailed subregion cluster information. First, let's sort the table by combined cluster size to find the sequences assigned to the largest cluster. We can sort by simply clicking the column header. It looks like the largest cluster has the cluster ID 18571, and it has 96,842 sequences, which is about 39% of the total sequence count. You can filter to include those 96,000 or so sequences by right-clicking the cluster ID, choosing Filter, and choosing to filter all values that equal this cluster ID. The total sequences left in the table now matches the cluster size. Note how the information about this particular cluster is copied to all sequences belonging to that cluster. Next, let's examine how those 96,000 or so sequences are broken down across the two samples by scrolling to the before and after counts columns. It looks like 1,596 sequences were found before panning, and 95,246 sequences were found after panning. That represents a 59.68 fold increase after panning. The counts have also been normalized and shown in the normalized counts columns. They are shown as a proportion of the total sequences per sample. The fold change is also calculated on the normalized counts. Let's remove the filter and explore the results even further. Let's say we want to find the total number of clusters, filter to remove clusters that have a low count, and isolate representative sequences from the top 30 clusters with the highest fold change. One sequence per cluster is chosen as the representative, so we can find the total number of clusters by filtering to include only the representative sequences, because we know there is only one representative per cluster. Start by typing part of the representative sequence column name in the filter. Select or press Tab to also complete the combined cluster _ representative column name. Select or press Tab again to choose the equal operator. Type true and press Enter. This will isolate all sequences with a tick in the cluster representative column. You can see from the total number of remaining sequences, there are about 28,000 representative sequences, and therefore about 28,000 clusters. Next, let's filter to include clusters that have counts of at least one in each sample. Type before to find the before counts column in the filter. Select before _ counts. Select or type greater than. Type 1 and press Enter, and do the same for after counts. That leaves 1,855 clusters. Then we can sort the table by fold change by clicking on the fold change column header. We can then shift click to select the top 30 sequences. You can see the number of selected sequences at the bottom of the table. We can extract those to a new document. This will copy the sequences to a new document, which we can then use as input for further analysis, such as multiple alignment. For more information about multiple sample comparison and biopanning, see our documentation pages on cluster multiple samples and identifying statistically enriched clusters within samples.